as you heard from Charlotte about what soil health is, how we define it, how we measure it. And so Charlotte Norris, our first speaker, joined us because we have such an honor to have Charlotte. She worked for the Soil Health Institute for several years, as you saw, implemented this massive cross-lab project that we're now reaping the rewards of, because we are getting such incredibly useful guidance on what measurements are useful, which are practical to deploy over a large number of samples like we'll be generating in this living lab. And Charlotte has come back to Canada and is now working for Natural Resources Canada at the Pacific Forestry Pacific Forestry Center. Yeah, thanks. In Victoria. So that's why I'm here today. That's why I was so thrilled to get to her talk.
Well, thank you, Kirsten. I want to talk about choosing soil health measurements for large scale adoption. I think in life, we have a lot of decisions to make. We have a lot of goals, and sometimes it can be which bicycle do I have to take to my favorite ice cream store. I use the same analogy when you're thinking about land management. There's a variety of different practices you have, and how do you get to your goal? My goal is soil health. There's a number of tools that we have in our toolbox to try and evaluate our practices and understand if we're getting towards our goal and how well we're getting there. In addition, I think it's really important that the tools that we have are intuitive to a wider audience. So if I said, we measured these different practices with a stopwatch, you kind of get how we're achieving our goal. So I think the tools when we're talking about land management and soil health need to be in the same sort of vein. They need to be easily understood. So tools aren't new. This has been talked about a lot. I was reminded of this. I taught intro soils to Vancouver Island University a few years ago, and digging around in the back of the lab cupboard, and I came across the Sudbury soil test kit, bigger and better crops. So really, this discussion about better crops, testing our soils, it's not new. It's really reminded of that. We talk about soil tilth or soil fertility, soil quality. Well, now we're talking about soil health. And I think that's the difference. I think we're now talking about soil health. And I think, to me, this is the goal. I'd argue with Kirsten about the title and being under co-benefits. I disagree. I think this is the goal, is soil health. And the definition here is that of the USDA NRCS, the National Resources Conservation Service, the capacity of the soil to function as a vital living ecosystem that sustains plants, animals, and humans. And it's the same definition that the Soil Health Institute uses. And it's become, I've seen it connects with people. This definition really resonates with a wide audience. They can connect with the idea of ecosystems, plants, and animals, and humans, and all those different components. Henry Jensen has a great definition out right now too, but it doesn't roll off the tongue quite the same way. And I'm sorry for that, because that one sort of gives context to soil health being important to where you are in the landscape. But this one is that used by NRCS, the Soil Health Institute, and the one that I'm working with in this talk. And to me, I took this picture by accident a few years ago, and it really tied in the different concepts to me, because you're capturing the idea of the humans, and the animals, and the plants, and all these important components. So what we're after, and I'm sure this audience knows, but I always like put up a picture that shows the functions of soil health. And so within that, like the Sudbury soil test kit was after, was food, fuel, and fiber. So that's really important. But what you're talking about here is also carbon sequestration. So you've got that component, where there's also water. It's really important in the water cycle, purification, regulation, erosion, habitat, nutrient cycling. So those are all the really important things that soil does for us, the ecosystem services. So those are our goals, but how do we get there? How do we measure how we're getting towards that goal? And so this was the idea of the project I was involved with before sitting where I am now at Natural Resources Canada. I was with the Soil Health Institute on their North American project to evaluate soil health measurements. And it was a crazy time. And I'm reminded here, it was about 4 years ago, I spoke to PRSSS, and we were just starting this. And so those who heard some of that, it was a blur for me. That was the beginning, and this is the end of the talk. And so for those who weren't there, I'll do some intro onto the project and then get into the results. So the North American project to evaluate soil health measurements, it's a large scale, broad assessment of soil health indicators, old ones and new ones across a wide range of soils, climates, management. So it's to get at the idea of sensitivity of widely applicable measurements to changes in soil condition from management practices. So really to get at which measurements would be best to measure our practices to know we're attaining our goal. So it's a measurement project, measurement. So yeah, there's a lot there. SHI convened a panel of experts, scientists, to say which one should we look at. And they said, well, here's about 30 core measurements we think are important and the methods that should be used associated with those measurements. And then so they looked at those in terms of tiers 1, 2, and 3. 1 being sort of methods and measurements we know a lot about and we know will be indicative of the condition and changes to management. And then 3 into 1 is where they show promise and they're more exploratory. So that's kind of the range of going from texture to genomics. So those are the ideas there. In addition to this core group of about 30 measurements, which, yeah, I think it comes in the package, so don't worry about the details in that. Or a whole suite of other measurements as well. So if people wanted to go in and to do some of the soil health indices that are out there, like the Haney test or the CASH, the Cornell assessment for soil health, those measurements are also available within the data set. So there's redundancy within the overall data set. So the data set comprises all those measurements done on over 2,000 samples. So those samples come from 120 sites, and the sites had to be at least 10 years old. The sites were associated with generally university, research facilities, and federal governments. It had to meet 5 criteria. It had to be physical disturbance, so that usually meant different rates of tillage, minimum till, no till versus conventional till, or cover crops, crop diversity, nutrient management. So that could be different rates of fertilizer, it could be compost, or it could be manure applications. Water management was another area of interest. And also location. So they really wanted to be able to have it at the continental scale. And so you have that diversity in climate and soil type. So I was the project scientist representative for Canada. And as a Canadian, I was sorry that we weren't able to capture anything from Atlantic Canada. So there are no samples from Atlantic Canada. And then as a British Columbian, I was really sorry that we didn't get any samples from BC. So that's unfortunate, but it was a really fast rollout through 2018 to 2019. I collected samples from April to June of 2019. The 9% at least of the samples were collected from mid-February to beginning of July of 2019. The remainder were collected in the fall of 2019. And that was to be able to collect our samples before any spring management activity occurred across the grain crops, intensive vegetable production, and the grazing, the pasture sort of sites. And then so the details on the Canadian sites recently came out in the Canadian Journal of Soil Science. And yeah, there's just so many samples. What I wanted to highlight was that each sample that we took becomes really hard to talk about samples when you're going across trials. And so we settled on experimental unit. So that's your each sort of a kind of a plot level discussion that you have. So each sample we talked about as an experimental unit that was split into 5. And so there were 5 labs across the US that handled all these measurements. And so the same lab handled the measurement. So for example, pH was done all at one lab, or genomics was handled by another lab. So that's just a little bit about the logistics of this project. Okay, some of the results. So the first paper that came out was by Diana in 2022, I believe, SSA. And that one was really reinforcing the importance of particle size analysis and the importance of, knowing the texture of your soil. To me, it was interesting. It was by the pipette method. I think reading about a slightly better accuracy than the hydrometer method that I know, but that fed into the pedal transfer function. Then you get into water and understanding water and the water cycle associated with the soil function and the importance of, just water retention and water cycling. So in addition to sort of the bulk samples that we took from these sites that went off for pH or texture, we took 2 cores. They were average for bulk density, and then 2 cores that were intact and shipped to the lab. And then, so those were measured on a tension table for field capacity. And that measurement came out as really important in being like sensitive to the site characteristics and management, and the details of that one and other measures related to the physical and the water cycle are in the SSAJ there by Diana. So when I talked about redundancy within the data set, that came out with the aggregates when you're measuring that. Because we know that the aggregates, they're so important that when you get into the soil functions again of water, the porosity, retention, erosion, or you could talk about in terms of carbon sequestration and relating it into maybe even habitat for organisms. So they're really important in overall soil health. And they were measured in 4 different ways. One was the Cornell Wet Aggregate Stability Test, or the rainfall simulator. Then there were 2 done by wet sieving. And then the 4th method was image recognition using the Slates app. And so that was sort of one of those tier 3 newer technologies that was investigated. And so all 4 of the metrics came out as being important that were good for measuring the site characteristics in response to management. So the aggregates were put out by Liz and Geoderma. And then from there, Dan did a really nice article highlighting the soil carbon and the carbon cycle in terms of soil health and soil functions. And he looked at soil organic carbon, the permanganate oxidizable carbon, POC, active carbon. Some people talk about it in terms of more labile or readily decomposable carbon. There was also the potential carbon mineralization. So that was a 24-hour burst test respiration and 96-hour as well. Beta glucosidase enzyme activity. So that's starting to tie in the microbial community and how well they're able to decompose what's present. And then the water extractable organic carbon, perhaps another labile carbon one. So again, you're getting at multiple soil functions in terms of carbon sequestration, nutrients, water management. Like it's just the carbon's really important that way, and it ties in so many different ways. And so what we have here, what Dan showed, was that as you'd expect, soil organic carbon, like that's it, that's the one that you want to have. And the best way to do it is by dry combustion. And then they also talk about the importance of, like Kristen talked about, the inorganic carbon, be able to pull that off. So Dan is working on the nitrogen paper, and all the results, it's a mirror of the carbon. So that should probably be out in the next year or so. Some of the biological measurements have been published. The PLFA paper came out in January. And so it identified differences with site characteristics and was able to pull out differences with different management practices. And same with the genomics data. Some of the genomics data was published in SPP by Liz. And it really showed that different tillage practices do influence the microbial community. You have different compositions and minimum versus intensive tillage systems. So again, we've got this whole suite of measurements that we're taking across many, many, many samples. And a lot of them are showing promise at being soil health indicators. But you need to think more in terms of, again, go back to the large scale adoption. And the objective of the project was to get it recommendations. And so it's not only can they show the effective management of the condition, but what's the availability of the test and what's the cost of the test. And so SHI reviewed the data thus far, and they published recently in Soil Security that they have the 3 measurements: total organic carbon by loss by dry combustion, aggregate stability, and 24-hour carbon mineralization. So it's with those 3 that you get at the idea of soil health. You get at so many of the different functions. You have the overall carbon quantity that's present, which links into the carbon sequestration and informs you of the nutrients cycling, which can help you understand the food, fuel, and fiber. And then it also gets into the aggregate stability. And this was by the Slate app. And so it's using that. It's not currently available at commercial labs, but it's easily implemented and ramped up. And so that starts to tie into all those other soil functions. And then we get to a carbon mineralization. So again, you're feeding into that. You've now brought in the biological component to interpreting the status of your soil. So those are the 3 measurements recommended by the Soil Health Institute. And Liz recently gave a great introduction to these 3 in a webinar online. And I believe it's now available on their website. So if you want to know more, it's there. And then I was going to transition to considerations. So those are the 3 offered by the SHI, total organic carbon, aggregate stability, and the 24-hour measurement. But so when you're choosing your measurements, what are some of the considerations? And so unfortunately, as I said, we weren't able to capture Atlantic Canada or British Columbia, but there is, when you're looking at it, you're trying to get this wide range of climate, and they used the Hargrave Climate Index, and that was a difference between evapotranspiration and precipitation. And so the greater the difference between evapotranspiration and precipitation in the map, it's red. And that's what we had more towards southern, the US and into Mexico. And the rest of it colors blue. There's less of a difference. And so if you see the picture, you start to see that the BC range for this climate index falls out throughout much of the sites for the project. And the same goes as well for the soil map. I showed where the sites are in relation to the FAO World Soil Map. And it's just, there's such a range of soils that were captured across this project that while it's, again, it's not represented in Atlantic Canada or with British Columbia, I think these indicators would still hold, because I'm so astonished after what I did see across the country that there is this consistent response. So I think we would see responses. Oh, and then it gets hard to see the dots on the map, but those are the ideas with those maps. It's just to say there's huge variability in climate and in soil type that were captured within this project. So I think while there weren't samples from British Columbia, the results would still hold true here and in Atlantic Canada. And the intent with this picture was also to show the variability in texture. I don't know why the image sometimes just doesn't work, but the texture was just across the board. They were from all categories captured. So again, that variability was present within the data set. So I think you would still have these indicators holding true. And so the other considerations I think you have for large scale adoptions are the methods. And so, yeah, I was nodding my head away to this morning's talks about picking your method and sticking with it. So as soil scientists, we can talk a lot about pH. It seems like a basic measurement. How are you going to do it? And I can vouch for that. There were conversations like this around the table when we were going through the project. And so are you going to do it in water? Or are you going to do it in calcium chloride? So once you decide what you're going to do, have the same lab do it. Because I think that's where some of the ability in this data set came through, was that the same lab measured pH, the same lab measured total organic carbon, the same lab did genomics. And so you remove the inter lab variability there. And I think that starts to feed into my interest in data availability. I think there's this huge interest in knowing more about soils across the country, across agriculture, across forestry. I mean, just what do we have? Where is it at currently? But there's also this interest not only in the breadth, but in the depth. How is soil responding to climate change, to forestry, harvesting, to fire, to insects, when I have my forestry hat on. And then in agriculture, cover cropping, reduced tillage practices, how is it responding? So you have that depth. And so making this data all available in a fair manner, so findable, accessible, interoperable, and then I always forget the last one, reusable. So those are there. And then my last point on there is lab capacity. I think that's a huge, it's just, yeah, I'm facing it now. The analytical lab associated with my research facility is pretty much at over capacity. The other research facilities associated with my organization in the same state. When you have the Senate and you have the House of Commons both talking about soil health, you have colleges developing curriculum for regenerative agriculture and soil health. I think this has only become like, to get there, you need to test, but who's going to do the testing? So I think as a community, that's going to be a really big issue, is who's doing that. I saw a great webinar by the Kootenai Boundary Farm Advisors. And within that, there was a nugget, somebody had to send their soils to Ontario. So where's the capacity to do testing? So I think within that, it's just the things that we need to think about when we're talking about how do we get to our goal? How do we get to our ice cream, our soil health? What practices are we using to get there? And what's the most obvious metric to measure it? And three of the ones that are coming out are total organic carbon, aggregate stability, and the mineralization. And the last slide was a thank you.